Hello, this is Dr. Paul Cottrell, and I am recording this on 5/9/2026 at approximately 12:49 p.m. This video is going to be focused on a research study that was published October of 2010 about investigating the Andes virus glycoproteins. Now, again, the Andes virus is the New World hantavirus that is been in the news due to that outbreak on the cruise ship. My understanding, there are 3 people that are dead and 5 additional cases as of this recording. Now, what this paper did was design a vector system, a lentiviral vector system. Now, lentiviral vectors are from the family of retroviruses, and lentiviral vectors are coming from viruses that are like SIV and HIV. So you have retroviruses, and then you have a genus underneath that retrovirus family called lentivirus virus, and then under that, then you have different viruses like HIV, SIV. All right. Now, so there is a connection between immune deficiency viruses and the Andes virus through research that's taking place. Now, they make these vectors on purpose to study some of the characteristics of infections of certain viruses, how they dock to a receptor, how they fuse, how they incorporate into the cell, and also some of the cellular mechanics. So building these vectors are important to understand the scientific aspects, but they can also be used for dual purpose. And those dual purposes can be nefarious, especially when you're talking about bio something like biological weapon development. All right, so this paper, published in October of 2010, developed a lentiviral vector system to study the role of the Andes virus glycoproteins. So these glycoproteins, I've done a few videos about them. They're the GN and the GC. And the Andes virus has a capability, most likely through zoonosis, but it has a capability to dock to the protocadherin 1 receptor that will allow for the Andes virus to get into the cell, this hantavirus to get into the cell and to propagate. And that the Andes virus can be clathrin dependent or independent, which is important. So this paper is more focused on building this lentiviral vector system. So let's go through the abstract. To infect target cells, envelope viruses use their viron surface proteins to direct cell attachment and subsequent entry via virus cell membrane fusion. How hantaviruses enter cells have been largely unexplored. Now, you have to remember, this was written in 2010. We know how they're done now. To study early steps of Andes virus cell infection, a lentiviral vector system was developed based on the simian immunodeficiency virus, SIV. All right. Now, this is the primate version of HIV, but they created what they call a vector pseudotype. So this has been modified in a way where it's not infectious and that it can be used for experimentation in the lab, typically in a BSL-1 or a BSL-3 lab. So they created this vector pseudotype with the ANDV. So this is the Andes virus, GNGC envelope glycoproteins. So what they did was they took this pseudotype vector, SIV, and added in the spike from the Andes virus. And that now is the spike for the SIV virus, the vector. All right. But you can see the dual purpose. So they have a pseudotype, SIV, which is the primate version of HIV. They mod that vector pseudotype is modified in a way where it's not going to spread, SIV, and they can do laboratory experiments. But by knowing that it can be done, you could easily in the laboratory create a non-pseudotyped SIV with the SNGC envelope. In other words, you can make an SIV virus instead of having its typical SIV glycoproteins spike. You have now modified and allowed for a chimera of Hantavirus Andes as a spike for SIV. Now that's not what they did in this paper, but this is the point I'm making with bioweapon development. When you are doing this type of research, you can go one step further without the safeguards and build these chimeras that could leak out of the laboratory. And this is the problem that I have with this type of research. But in this paper, they used a pseudotype that is safe to work with. But researchers can basically take those guardrails out and modify in a BSL-4 lab and incorporate the GNGC glycoproteins into SIV or HIV. And that's the problem that people need to start waking up. When I keep on saying for the last 6 years that there needs to be more regulation on this type of research. So I'm going to continue here. The incorporation of GN and GC onto the SIV-derived vector particles was assessed using newly generated monoclonal antibodies against Andes glycoproteins. In addition, sera of the Andes virus infected humans were able to block cell entry of the SIV vector pseudotype with the Andes virus glycoproteins, suggesting that their antigenic conformation is similar to that in the native virus. So now you can see why it's important to do the pseudotype vector system for SIV to test potential therapies, either antibody, some sort of antiviral medication that will help to block it, or monoclonal antibodies. There's lots of research that can be done with these pseudotype vectors. But if you take the guardrails out, you can just go one step further and just incorporate GNGC right in to a native SIV virus. And now you have a chimera that it has gain of function where the SIV is now attaching to the protocadherin 1 receptor, which can be a problem. And you can do that with many different types of viruses. And this is the reason why I keep on saying you got to pay attention to the 4th and 5th cocktail of SARS and what they've been doing in the laboratory. So, but in this research study, they just used a guardrailed SIV called a pseudotyped vector, right? But you can imagine not doing that and just incorporating the GNGC into the SIV. And you can do that by infecting a cell with SIV native and have uh, a either vector system that's incorporating the gene expression of GNGC, or through a plasmid, and through a random recombination over time, it will combine. But better yet, you can actually just build it by just using plasmids and have a plasmid that codes for the SIV, take out the SIV take out the SIV typical glycoprotein spike, and insert the code for the GNGC. But the point I'm making here is that you can put on an SIV or an HIV GNGC. That's the main point I'm making here. All right. But in this paper, they didn't do that. All right. Now, so they create this safe vector, so pseudotype vector, SIV, with the Andes spike, the GNGC, and they were able to test to see if it can be blocked. And it shows that it can. Now let me continue reading here. The use of such SIV vector pseudotype with the Andes virus GNGC glycoproteins should facilitate studies on the Andes virus cell entry. Along this line, it was found that depletion of cholesterol from the target cells strongly diminished cell infection, indicating a possible role of lipid rafts in Andes virus cell entry. Now, this is important because there's research, strong research, that shows that the primarily the GN of the spike, attaches to the F83 and D85 area of the protocadherin 1 receptor in the location of the EC1. Now, these lipid rafts are important on the cell surface, where it kind of it combines certain types of receptors together, gets them closer together, and it allow for fusion of certain capability, especially for a virus. So if a lipid raft is coming by, it might have the integrins on the lipid raft that actually helps with the fusion. Now, there's been some studies that are looking at I think it's the beta-3 integrin that helps the Andes virus, the Hantavirus, New World, to bind to the cell. All right. So you have the GN that can attack, that has been that in the New World been modified through just mutations in nature, now attaching to the protocadherin 1 EC1 domain. And it also seems to need a helper, and that helper seems to be a beta-3 integrin. Now, the lipid raft may be helping either add and consolidate the protocadherin 1s and the beta-3 integrins together. So if you get rid of the cholesterol that helps to build up the lipid raft on the cell surface, then it's harder for the Andes virus to get in. But if you were losing if you were losing your cholesterol in your cells, then you got a lot of other problems too if you're losing your lipid. But this research here could be part of a potential antiviral therapy where you target the surface of the hantavirus, maybe with a monoclonal. You can also target with maybe blocking production of lipid rafts using some sort of medication as a potential other way to block viruses. A multi-therapy method is much better than a monotherapy method with viruses because of the mutation rate and the production rate of viruses. They have a tendency to emerge a method away from the therapy. But if you lock it in by using maybe 3 different methods, then it's blocked and it stays contained and doesn't drift too much. But if you have monotherapy, viruses will mutate and drift away from the therapy. So it's potential to have some sort of medication that helps block the lipid rafts, but I see this as a very high potential of side effects if that's the case. But it's interesting to realize that it seems that the lipid raft is part of the helping mechanism of the cell to fuse with the hantavirus, especially with Andes. All right, I'll continue here. The GNGC pseudotype SIV vector has several advantages, notably high titer vector production, so it can produce in the cell at a high rate, and easy quantification of cell infection by monitoring the GFP reporter gene, expression by flow cytometry. Such pseudo SIV vectors can be used to identify functional domains in the GNGC glycoproteins and to screen for potential hantavirus cell entry inhibitors. So there is a benefit of using the pseudotype vector from a lentivirus like SIV. But I want to make you understand that you can put the GNGC receptor of Andes on HIV or SIV. That's not pseudotyped. And that's dangerous. That's like super dangerous. So if you want to gain function of another type of receptor for HIV, you can just plug GNGC right to it. So my point here is that we need a little bit more of a discussion in the public domain about the regulation of these types of research and the safety protocols. Because a lot of times what happens is they take a dangerous virus that's in a BSL-3 or BSL-4 category, and they downgrade it by taking pieces of it and putting it into a pseudo vector, and then now they're doing it in a BSL-2. And maybe what we should be doing is making sure that these types of research be maintained at a BSL-3 or a BSL-4 laboratory. Maybe that's one way to regulate this. But again, this research has dual purpose. You can make weapon systems out of this if you're smart enough. So this gives you a little bit of a take of what's going on with the Andes and what was happening in 2010. That they made a pseudo vector using SIV. And that with the technology we have today and the knowledge base that's out there in the world, you can make a weapon out of this. I'm not saying it happened, but it can be done. With that, Please help support the work that I do by making sure that you subscribe to all my channels. All the links are in the description of this video. If you would like to help support the content, please be a paid member to my Patreon channel or my Substack channel. You can also donate on my website, the-studio-reykjavik.com. You can donate through Stripe, PayPal, or buy me a coffee and take a look at the great products that I offer. I do all these videos for free. It takes quite a bit of time for me to digest this information and boil it down into understandable pieces for individuals that are not necessarily from a medical or a scientific background. Thank you for listening and have a nice day.